OK，好，大家好，我们今天的 topic 是 immunology，然后由我讲 introduction，然后武艺讲research，然后呃另一位朋友我不知道他的英文名，但是他是负责讲 industry，然后哦然后我找到的一篇review paper，然后it's 非常 sorrow 的讲了，嗯，like what basically very sorrow introduction to immunology and immunopathology. 所以it's gonna sounds like a mini lecture, but um, actually, if you're not familiar with immunology, you could get a lot of from that paper. 嗯，然后 so for our immune systems, we have basically two separate uh, like immune system. We have innate immunity and we have adaptive immunity. And then I'm gonna start with innate. That's like our first line of defense. It's a very rapid, quick response, and it relies on pathogen recognition receptors, and which will recognize pathogens that have uh, molecular common structures, which is called PAMs. So once those receptors bind to the structures, it will induce a series of response, including rapid recruitment of immune cells uh, to that like injury site and site of infection, and will also induce inflammation through cytokines and chemokines. And so the immune cells, which I'm going to talk about later, but basically it just like phagocytes, like macrophages, neutrophils, like those cell types. And then like one of the most important things in innate immunity we have to talk about is the complement system. Uh, Basically, it's a pathway that will make pathogens susceptible to phagocytosis, and it will activate adaptive immune systems through mobilization of antigen-presenting cells. So basically, everything is to like induce inflammation to recruit more body fluids, which will bring more phagocytes. We want more phagocytosis to eat all those infected cells and pathogens. So on the right, it's a very detailed pathway of complement system. Um, Basically, it will like the end of the pathway. It's phagocytosis and inflammation, and it will cause apoptosis. So basically, it's just a way of recognize and kill the infected cells. So on the left, it's a very inclusive chart of what kind of immune cells are involved and what do they look like, where do they reside, and what kind of targets they're mainly responsible for. And also different phagocytes, they have different um, lifetime lifespan. So for macrophages, it's relatively long, but for neutrophils, it's really short. Um, and also for example, for mast cells, uh, they reside in tissues, but for basophils, they are mainly reside in circulation, your bloodstream. So that was like just give you like an idea of what's innate immunity, and there's obviously like more than that. And it's also included in the paper, so I really recommend reading that paper. So going off to adaptive immunity, which is the immune system that we're more familiar with, I guess. It's the basis of effective immunization against infectious disease. So that so adaptive immunity come to place when innate immunity is not enough to kill all the pathogens. So what adaptive immunity do is they will recognize and distinguish non-self antigens from self antigens. So they will activate the pathogen-specific pathways to eliminate pathogens, and they will also develop immunological memory. So that's the rationale we utilize to make vaccine. And two main cell types that are involved in this process are T cells and B cells. So like really briefly, that T cells will be activated to proliferate through actions of antigen presenting cells. So they need that antigen presenting cells to bind to for the T cell receptor to bind to antigen. And each T cell receptor is specific to a specific antigen. So it's like one-to-one. And B cells, they're kind of similar but different is that they don't need that antigen presenting cells to bind to antigen. They can bind directly. And once bind to that antigen, they will differentiate to plasma cells to produce antibodies. And they will, other than plasma cells, they will also differentiate to memory B cells. So on the right, it's also like a very, like it gives more information about what could happen like after T cells bind to the pathogens. But basically, it will produce antibodies after bind to B cells and recruit like immune cells after like macrophages and killer T cells. So that's like very, very, very brief and summary of adaptive immunity. And going off of that, I'm going to talk about, so like within adaptive immunity, what are like two different types of mediated response. So we have antibody mediated and cell mediated. So antibody mediated, we talk about that it's cells, they secrete cytokines to help B cell multiply and produce antibodies, which will, once they bind to antigens, they will recruit like phagocytes to eat the antigens and whatever they're flagged. And there are different antibody classes that I have a chart and a diagram on the right showing like they're categorized by different functions, and they also reside like different like in different parts of the body. So for example, IgM is the first antibody that will be produced when you first encounter the antigen. And once we develop that memory, if you encounter that pathogen again, it will be IgG that's produced, and that will be more specific and rapid. So besides antibody mediated, we have cell mediated response. So that's mainly defend us against intracellular pathogens through cytotoxic T cells, macrophages, NK cells, like natural killer cells, and cytokine production. So cytotoxic T cells will induce apoptosis in cells displaying foreign antigens, and macrophages and NK cells will destroy intracellular pathogens, and it will also stimulate cytokine production. So this is also the response to target like cancer cells. And then after the like rationale of the immune systems, I want to talk about what kind of like pathologies we have, like what kind of malfunction, what could go wrong to our immune systems, and what would happen. So basically, the paper talked about like hypersensitivity, which is our immune system like overwork itself, like it will produce like undesirable response by like a normal immune system like stimulus. For example, allergy. It will like the immune system will produce a very like very strong reaction toward the allergens. And then autoimmunity is like a not normal response. So like our immune system will attack our own cells or our own tissues. So it's often marked by self-reactive T cells, autoantibodies, and inflammation. So they gave examples of like celiac disease, which is when you are allergic to gluten, and also type 1 diabetes, which is when your immune system attack your own beta cells. So it can no longer produce insulin. And inflammation is when you constantly have like you're inflamed, you have chronic inflammation, which is poorly regulated inflammatory response and tissue damage. So we want inflammation for a short period of time to recruit all the cells we need, all the fluid we need. But once the inflammation is chronic, it's long term, it will damage the tissue. So, for example, arthritis, psoriasis, and asthma. So psoriasis is like a skin condition where you constantly have inflammation and have like a rash. So, and also we have immunodeficiency, and that is when our immune system is compromised and we could lose like partial or entire response, and that could be caused by genetics and could also have like secondary cause like environmental factors such as bacterial, viral infections, and also can be caused by malnutrition, and can also caused by, for example, if you have autoimmune disease, which is which us talk about like your immune system attack your own tissue. When you take drugs to suppress that, like when you try to suppress your immune system, if the patient does not respond to that drug or respond to that drug too well, it will suppress too much of your immune system. So AIDS would be like a very famous example for a viral infection that's caused by HIV. And I think that's it for introduction. Does anyone have any question, or do we take questions afterward?
Yeah, we can take questions at the very end. So I'm gonna share my screen. Okay, this one. Okay. So I'll be presenting this paper, which is called The Age Tumor Microenvironment Limits T Cell Control of Cancer. So T cell is a very important immune cell that basically kills all the cancer cells and also the cells that got infected by virus and stuff. So the researchers want to know that if aged microenvironment actually limits the T cells function. So the inspiration comes from that the researchers inject both melanoma cells and this lung carcinoma cells into mouse from different ages. And they found that in older mouse, the tumor actually grow faster and grow into higher volume, so that they want to know if this microenvironment actually affects the immune cells. Yes. Okay, and then they get the cells from all different ages and then do a single cell RNA sequencing. They found different cell populations. And the young and age mouse actually have different cell populations. So in young mice, they mainly have what they call T term and T progenitor. You can see here. Progenitor is progenitor-like exhausted CD8 plus T cells, and T term is terminally exhausted T cells. Just for information, exhausted T cells are T cells that have already functioned and lose their ability to kill cells. This is actually good because in this way, it means that they are function like correctly in their way and they're doing their job. And in aged mice, the more enriched cell population are less differentiated T cell states, like naive-like T cell, memory-like T cell, and transitory T cells. They're less differentiated and I guess worse in doing their job. And they also identify a unique cluster of T cells that is enriched in aged tumors, but not in young tumors. And this cell state has not been previously characterized, so they think this is a unique cluster, which is tumor infiltrating and age associated. So they call it T-TAT cells and want to dive deeper to investigate these cells. Okay, so first they want to know that if these if the aged mice have if the T cells in aged mice are genetically defect, so they call it cell intrinsic defects, so that they can do their job. So they do this by obtaining both T cells from young and aged mice. The young mice T cells are in blue and the aged T cells are in red, and they injected into both of them into young mice to give them the same tumor microenvironment to see if that makes any difference. And this is the result they get. From this figure, they figure out two things. The first one is that even from the earliest phases of T cell infiltration to tumors, which is day 7, aged OT1 cells have a reduced ability to form T progenitor-like cells, which is like the good cell. And also, as the days go on, the ratio of T terminal and T progressive progenitor-like cells grew further, which means that the aged OT1 cell, the aged mice have poor ability to maintain T progenitor-like cells, ultimately promoting premature terminal differentiate in the TME. And just a few tips on reading this graph, because I really struggle on it. So on the vertical axis, the SLAM6 is a marker for T progenitor-like cells, and on the horizontal axis, the marker gene actually mark the terminally exhausted T cells. And the number here should add up to 100, and it gives the 4-quarter gives you like how many % of a specific cell population is in each quarter. Okay, and then they want to see that like what other stuff does this cell intrinsic characteristic do? So they realize that the cell intrinsic defects in aging promote premature T cell differentiation and provide genotoxic stress, but they do not drive the differentiation of T-TAT cell subset in the HTME. So that there must be other things, which is they later find out to be extrinsic signals that actually drive the T-TAT cell state. So to investigate how the extrinsic signals affect the cell state, they do the right opposite than the intrinsic experiment, which means that they only get the young OT cells, and then they give the young OT cells different microenvironment. So they inject it into both young and age tumor-bearing mice, and then to see if that makes a difference. So yeah, this is really complicated. So I'm just gonna read out the conclusion. So age mice has a higher proportion of T-TAT cells in their tumors, and then yeah, it experienced worse tumor control due to their poor cytotoxicity. So actually, only the mice that get T progenitor-like cells has somewhat help with controlling the tumor, and all the others are not. All the other T cells are not helping.
I have a quick question.
Yes.
So for this slide, you said T-TAT cells in their tumors experience worse tumor control due to their poor cytotoxicity. 所以 like are these T cells supposed to have cytotoxicity? Like how does cytotoxicity plays a role in this?
对，就是T cell are supposed to have a role of cytotoxicity，因为他们要杀死不好的细胞嘛。
Okay,
但是
okay.
对，
That's
但是
what-
这个 T-TAT cells 呢，它是在 aged mice 里面发现的一个新的 cell population，所以呢，这个 researcher 就发现他们其实是没有就是正常的 cell toxicity，也就是意味着他们没有办法 play their role and do their job into killing like the correct tumor cells。
Okay, 好的，谢谢。
对，然后呢，他们就找了一个 human cancer 的data set, and then look into that to see if they can also find the T-TAT cell population, and they actually find it, and which is pretty surprising that also in tumor biopsy from older individuals, they have more T-TAT cell population, but not in the younger individuals. So this part is really important for the whole paper. They found that T-TAT cells represent a cell lineage with reduced effector capacity that is distinct from T cell exhaustion and is also expanded within the tumors of older individuals with cancer. 也就是说，T cell exhaustion 是在 younger individuals 里面有的，也就是那些已经做了自己的工作，所以失去了能力的 T cell。但是这个 T-TAT cell 呢，它是没有办法做自己工作的。然后呢，它是有 reduced effector capacity，所以呢，这两个是不一样的。他就是想去证明，这个他们发现的这个新的cell population 是在 aged group 里面特有的。And also, they found that the trajectory of CD plus T cell differentiation in general and the formation of T-TAT cells in particular are almost entirely driven by cell extrinsic signals in the HTME. 然后他们就想知道，到底是什么东西，到底是什么 extrinsic signals 在影响这个T cell 的performance and also what drives the T cell to become T-TAT cell。然后他们就发现呢， tumors in aged mice specifically has fewer interactions between NK cells, which is natural killer cells, CDC1 are dendritic cells, and T cells。就是说，这些细胞之间的crosstalking，就是它们的细胞通路断了，是因为年龄的增长而断了。然后呢，他们就是因为他们知道 dendritic cells recruitment are really important, and then they further dive into the dendritic cell population to see like what's happening. So they found that the decrease in dendritic cells and their diminished antigen presentation capacity may constrain T-term cell formation and facilitate TAT cell formation. 然后呢，最后一个 part as a conclusion is to test some therapy that could rescue the poor T cell priming by aged dendritic cells. So the first therapy they try is a carrier. They optimize a like mRNA delivery system using lipid nanoparticles, but they end up realize that this nanoparticle only helps the young mice group, which is probably because of lack local guidance from CDC1 cells in age lymph nodes and tumors. Yeah, 所以就是这个 tumors 这个 vaccine 就没有办法得到很好的 response. And the second therapy they try is a very famous one called CD40 agonist，就是 CD40 拮抗。然后他们发现这个就是能够帮助 restoring CD1 numbers with myeloid targeted immunotherapies，然后就是能够帮助CD8 T cell expansion, prevent TAT cell formation, and ultimately improve tumor control. Yeah, that's the end. 就是这篇文章它的整个 idea 其实挺简单的，其实就是想看一下 T cell 在 aged environment 里面会不会 lose its function。但是呢，它 end up using a lot of fancy techniques like single cell RNA, and also like it's really comprehensive because they test like two intrinsic like states, and also and then after the intrinsic experiments they come to the extrinsic experiments. So I guess that's why they get it published to Nature Immunology. Yeah. Okay, hope you enjoy it. Can go to the next part.
Um, 所以我的部分是immunology related industries. So first I'm going to introduce what are some key areas in the immunology industry. Some major areas include cancer immunotherapy, autoimmune disease treatments, and vaccines that are either preventive or therapeutic. And the development of antibodies will be really useful in the industry as well. So cancer immunotherapy has the goal to fight cancer by making some genetic modification to human cells so that it can target for tumor cells and then kill it, or like make some checkpoint inhibitors so it can stop cells from proliferate too much. And autoimmune disease treatments includes many inhibitors that stops autoimmune system to overreact. And then vaccines will be using some of the genetic information to target some disease, for example, the virus we get from COVID, and some other therapeutic vaccines can be used to stop immune system from attacking your cells tissues. And other antibodies can be like inhibitors or just labels that could be really helpful in the research areas. So I compiled some of the companies and some of their famous drugs. You can refer to under each category. I will not go through them and just give you an idea of how, like, what's their subject. Like you can see here, so under cancer immunotherapy, they like to focusing on inhibiting PD-L1 checkpoint, and then they will use a lot of CAR T cell therapy, and they will also design lots of monoclonal antibody that inhibits some types of receptors. And yes, and so if you're interested in some of them, you can go look at them. And then for autoimmune disease treatments, large bio companies like to treat IL, which is the cytokines secreted by T cells. So it can stop your immune system to overreact and causing inflammations. And lastly, the vaccines are really like familiar to us now because of the COVID. So they're mRNA vaccines or like normal vaccines that can treat infectious disease. For example, like HPV are designed within the immunology industry. And then, so here are other uses for designing antibodies or utilizing immunology to facilitate our like research. For example, like antibiotics that can help with immune assays and flow cytometry that can label different cells or biomarkers. It will be like really helpful. And then in next generation sequencing and immunohistochemistry will also utilize a lot of different kinds of antibodies that binds to specific antigens. So now I will introduce a specific disease and a specific drug that treats the disease. That's a really good example how biopharmaceutical companies will be like just a good example of how biopharmaceutical companies like make profit and then designing a very effective drug for arthritis, rheumatoid arthritis. So so this drug Humira, it is drug that's for treating autoimmune disease, rheumatoid arthritis. It is the immune system is overacting, and then it causes your joints to the mucous membrane like grow thicker and thicker, and the cartilage will like degrade. So it will cause your joints to swell and be stiff, and it will be really painful when you move it. So it's caused by the overproduction of pro-inflammatory cytokines, mainly tumor necrosis factor alpha, interleukin 1 and interleukin 6. And so the drug itself, it is called adalimumab. It is a monoclonal antibody. It originally was named D2E7 before it came out to be a drug on the market. And then it combines to the tumor necrosis factor alpha and stop the pro-inflammatory reaction triggered by these cytokines. And it the Humira stands for human monoclonal antibody. So human monoclonal antibody was designed by expressing a full variety of nucleic acid sequencing that encodes for different types of antibodies in virus. So they can display those antibodies will display on the cell surface, and then they will flow through these different types of cells with different types of randomized antibodies on top of the TNF alpha, and they will see which one binds to it. So they select the ones that binds the tightest, and then it becomes the adalimumab. So they don't really know why is it binding so good, but they know it binds well. They don't know the mechanism of like if the antibody binding with the tumor necrosis factor when they first designed this drug. So here is a more detailed diagram of the pathogenesis of rheumatoid arthritis. So there will be some trigger that promotes the inflammation in synovial membrane, and it can be genetic trigger or some like stress related trigger, anything. It's like unknown, but it will cause you to have inflammation in your synovial membrane, and it can attract leukocytes into the tissue. So the leukocytes will like secrete cytokines, and they can induce cytokines like TNF alpha and other IL factors that can induce MMP and RANK ligand. So MMP will cause the osteoclast, which is the degeneration of bones, and RANK will cause the degeneration of bones, and MMP will cause the cells to migrate, and the cartilage will like grow more. And when the adalimumab introduced into your body, it can bind to the TNF alpha, and it can stop the TNF alpha from binding into its receptor, so the inflammatory response can be interrupted, and so no more inflammatory actions will happen at your joints. So it downregulates the expression and stop everything from overreacting. And the half-life of adalimumab is like 10 to 12 days. It is longer than other non-human antibody. So Humira is one of the best-selling drug. It make profit of like $200 billion. So like why is it so successful? So of course their drug, their antibody is great. They can they have a good half-life. They can be like injected once a week, and it doesn't require physicians to do the injection for you. So it's convenient and easy to use. And moreover, the company, which is Abbott, it is the boss of their company's name. So Abbott provide Medicare patients with access to adalimumab. So at that time, self-injection medicines are not covered by Medicare. So Abbott launched the Humira Medicare Assistance Program to provide free Humira injections for qualifying patients until the Medicare drug benefit is covering Humira. And their boss about is really smart. They invested in clinical trials for a wide variety of disease. So because of the pathway of PNF alpha can be infecting lots of different autoimmune disease. So they invest in clinical trials that examine the Humira's effectiveness in other autoimmune disease. So they have their drugs covering a lot of disease, and it can earn money from prescribing Humira for a wide variety of patients, not limited to rheumatoid arthritis. And they also invested in long-term clinical trials. So lots of other drugs, they don't do that because it's money costing. But like these kind of drug will like slow down your like immune system, and it can cause some life-threatening infections, and it can cause some issues with your cells. It may incur cancers. So people are like concerned about the long-term effect of this type of drugs. So they invested in clinical trials that like trace patients for a long time range, for example, like 8 years of clinical trials. And they do a multiple of those long-term clinical trials, and to show people or like their patients it is safe, and it has efficacy over a wide range of time. And the figure on the right is just a figure showing the market size of Humira. It is the dark purple portion of those bar graphs. So it's like taking a lot of the market share, and then they're earning a lot of money because of this. So that's everything. Thank you for listening. If you have any questions, please ask.
谢谢子金。Aletta，要不要先把 recording 停了，然后我们可以再问问题，大家。
Okay.